In 2005, scientists announced something that happens only a handful of times in an entire generation of mammal research. The discovery of a species so unlike anything else alive that it needed its own brand new family placed on the mammal family tree, a taxonomic rank sitting above genus and species that most newly discovered animals never come close to earning, no matter how unusual they turn out to be. Within a year, a second team proved that announcement was wrong in the most unexpected way imaginable. The animal wasn't new at all. It was the last known survivor of a family declared extinct 11 million years ago, quietly alive the entire time in the limestone hills of Laos, while the rest of the scientific world assumed its entire lineage had died out before the first humans ever existed anywhere on Earth. Start with how the animal was found in the first place, because nobody set out looking for a living fossil, and the discovery itself happened almost entirely by accident. Robert Timmins, a field biologist working with the Wildlife Conservation Society, came across the specimens at a hunter's market in a small town near the Mekong River in central Laos, where local hunters sold smoked and freshly killed wild game for food to nearby villages. Among the usual bushmeat laid out for sale, Timmins noticed something he didn't recognize at all: a small, rat-sized animal with a long, bushy, squirrel-like tail, long whiskers, and a build that didn't match any rodent he knew from the region despite years of field experience across Southeast Asia. Locals called it kha-nyou, meaning roughly rock rat, describing where it lived among the crevices of the area's steep limestone hills that dominate much of the surrounding countryside, terrain so rugged and difficult to access on foot that much of it had never been thoroughly surveyed by outside biologists before Timmins happened to pass through the market that particular day. No live specimen had ever been collected or documented at that point, only the dead and smoked animals already sitting on market tables, which made confirming exactly what the animal actually was considerably harder than it would have been with a living individual available to observe and study directly in its natural habitat. Researchers Paulina Jenkins at the Natural History Museum in London and C. William Kilpatrick at the University of Vermont took on the task of identifying it properly, combining detailed skeletal examination of the skull and teeth with genetic analysis of the mitochondrial DNA extracted from the specimens Timmins had collected at the market. Publishing their findings in April of 2005 in the journal Systematics and Biodiversity, the team, crediting Jenkins, Kilpatrick, mammalogist Mark Robinson, and Timmins himself as co-authors, concluded the animal was different enough from every other living rodent, based on both its skull structure and its mitochondrial DNA sequence, to warrant an entirely new family of its own, Laonastidae. They gave it the scientific name Laonastes aenigmamus, roughly translating to stone-dwelling enigmatic mouse, a name that captured both where it lived and how genuinely puzzling its exact identity still was even after the formal description had been completed and published. Discovering a new mammal family is a rare enough event in modern zoology that it made headlines around the world almost immediately, treated by much of the scientific press as one of the more significant zoological finds of the entire decade, a genuine rarity in an era when most newly described species turn out to be relatively minor variations within families already well documented and understood. If you're finding this interesting, be sure to subscribe to understand more DNA from the past. The correction arrived less than a year later, and it came from an entirely different direction than anyone studying the living animal had originally considered. In March of 2006, Mary Dawson, Curator Emerita of Vertebrate Paleontology at the Carnegie Museum of Natural History, and a researcher with decades of experience studying fossil rodents specifically, published a paper in the journal Science directly challenging the new family classification, working not from fresh genetic data, but from careful fossil expertise instead. Comparing the rock rat's skull and dental structure directly against the actual fossilized remains of an extinct rodent family called Diatomyidae, a group known only from bones and teeth, recovered at sites scattered across Asia and presumed extinct since the late Miocene, roughly 11 million years ago, a group Dawson herself had spent much of her career studying through fossil remains alone, Dawson found the match unmistakable and immediate. The living animal from that Laotian hunter's market wasn't a new family at all. It was a Diatomyid, the very same ancient lineage the fossil record had shown nothing of for 11 million years, suddenly and quietly still alive in the present day, making it a genuine example of what researchers call a Lazarus taxon, a species that reappears long after being confidently presumed gone for good by an entire field of scientists working from an incomplete fossil record. The reason the original genetic analysis had missed this connection entirely came down to a limitation. No amount of careful lab work could have solved on its own, no matter how rigorously the original team had approached the problem. Genetic material from the actual fossilized Diatomyidae specimens had long since broken down completely, far too old and far too degraded after 11 million years underground to yield any usable DNA for direct comparison against the living animal. That meant Jenkins and Kilpatrick's original 2005 genetic analysis could only ever compare the living rock rat's DNA against other currently living rodent families. Since no ancient diatomide genetic material existed anywhere on Earth for anyone to test against directly. Without that missing piece of the puzzle, the genetic data alone genuinely supported treating the animal as something unprecedented, rather than a survivor from one specific extinct lineage, a reasonable conclusion given the tools and comparison material available at the time, but one that Dawson's direct fossil comparison method wasn't limited by in the same way at all. Subscribe to not miss the next one. Resolving the disagreement between the original genetic study and Dawson's fossil-based correction required an entirely new approach, one built specifically to test both competing conclusions against each other directly rather than simply accepting either interpretation at face value. In 2007, a research team led by geneticist Dorothée Huchon published a study in the Proceedings of the National Academy of Sciences, conducting a far larger and more comprehensive molecular analysis than the original 2005 study had attempted, drawing on 5.5 kilobases of sequence data pulled from 4 separate nuclear genes and 2 mitochondrial genes, spanning representatives of every major rodent group available for direct comparison across the entire order. The team also examined short interspersed elements, small, distinctive fragments of genetic code, sometimes called molecular fossils because they insert themselves into a genome at a specific point in evolutionary history and remain there permanently afterward, essentially unchanged, passed down intact to every descendant of the animal that first acquired them, offering an additional, entirely independent line of evidence separate from standard DNA sequencing methods and far less prone to certain kinds of statistical error that can sometimes complicate straightforward sequence comparisons. Every piece of that expanded genetic analysis agreed completely with Dawson's fossil-based tree, rather than the original 2005 classification, a consistency that held up across multiple independent genetic markers, rather than resting on any single data point that could later be second-guessed or reinterpreted differently by another research team. Using statistical tests specifically designed to directly compare competing evolutionary hypotheses against each other and calculate which one the actual data supported more strongly, the team formally rejected the new family hypothesis in favor of Dawson's conclusion, closing the debate with a level of confidence the original 2005 study alone had never been in a position to provide. The kha-nyou wasn't a scientific first after all. It was proof that an entire rodent family missing from the fossil record for 11 million years had never actually gone anywhere at all, surviving quietly in a small corner of Southeast Asia the entire time. That resolution reshaped how researchers understood the animal's place in the world, well beyond simply settling an academic disagreement over which journal published the correct classification first. The Laotian rock rat is now recognized as the sole surviving member of Diatomyidae, closely related to a small group of living rodents called gondis found today in North Africa. its closest relatives among any animal alive on the planet, despite an evolutionary gap stretching back millions of years since the two lineages last shared a common ancestor. Field surveys conducted since the initial discovery have confirmed the species living exclusively in a limestone karst region of central Laos, sheltering in the crevices and boulder fields at the base of steep, forested limestone towers at elevations ranging up to roughly 850 meters above sea level, a highly specific type of terrain found in relatively few places anywhere in the region. In 2011, researchers confirmed a second population living just across the border in Vietnam's Phong Nha Ke Bang National Park, expanding the species' known range to a confirmed total of roughly 150 square kilometers of suitable karst habitat, though that range remains tightly restricted to this one specific type of limestone terrain and nowhere else documented on Earth so far. The animal turns out to be omnivorous, feeding on the leaves, buds, fruits, and roots of at least 18 different plant species found throughout its forest habitat, along with occasional insects including cicadas, mantises, and grasshoppers, a genuinely varied diet for an animal that spent well over a century completely unknown to Western science, despite apparently having lived alongside local hunters in the same limestone hills the entire time without anyone thinking to formally document it. Confirming what the living animal actually looked like, rather than relying solely on the smoked and dead specimens Timmins had first encountered at the market, took additional fieldwork in the years that followed the initial discovery. Researchers eventually documented and photographed live individuals directly in their natural karst habitat, providing clear confirmation that the species was genuinely thriving in the wild, rather than existing only as scattered remains passed between hunters and scientists. That confirmation mattered enormously for conservation planning, since a species this restricted in range, confined almost entirely to one narrow band of limestone terrain straddling the Laos-Vietnam border, faces real vulnerability from habitat disturbance, hunting pressure, and the kind of localized environmental change that could threaten an isolated population far more severely than it would threaten a species spread across a wider geographic area. The International Union for Conservation of Nature currently lists the species as least concern overall, though researchers studying the smaller Vietnamese population specifically have flagged more serious localized concerns, given how tightly restricted that particular population's range actually is compared to the species as a whole. Line up the full sequence of events, and the Laotian rock rat story runs in almost the exact opposite direction of most animals that make headlines for their DNA. It wasn't declared extinct and then found alive the way a coelacanth or a quagga eventually was, rediscovered after science had already given up on it entirely. It was declared something entirely new, celebrated briefly as one of the rarest events in mammalian taxonomy, a species so unlike anything else alive that an entire new family had to be built just to hold it, before a second team, working from a completely different type of evidence, proved that celebration had gotten the actual story backward from the very beginning. The kha-nyou was never new. It was old, older than almost anything else walking around today, the last living thread connecting the modern world directly back to a rodent family. The fossil record had already written off as gone for 11 million years. Sitting quietly in a corner of the world, nobody had bothered to look closely enough to notice until a hunter's market stall in a small Laotian town happened to put it directly in front of the one scientist, paying close enough attention to realize something didn't quite fit.